For week 7, case study 7, I'm going to be talking about fluorouracil and the effects it has on DNA and RNA synthesis. So first, a little more about the drug. It is a heterocyclic aromatic organic compound. It's used as an anti-metabolite drug, and it interferes with the nucleoside metabolism, and it's incorporated into RNA and DNA, and then it is inhibited, which leads to cell death. So fluorouracil works as an inhibitor of the cellular thymidylate synthase, and it leads to the prevention of DNA replication. It also works by inhibiting RNA synthesis by the metabolites intervening with RNA. So to further discuss thymidylate synthase a little more, it's a folate, and it's a dependent enzyme that catalyzes the conversion of deoxyuridate monophosphate, DUMP, to deoxythymidine monophosphate, DTMP. And it's one of the nucleotides in DNA. It leads to DNA damage due to an increased level of DUMP. It functions as an RNA binding protein, and it interacts with p53 tumor suppressor gene. So it works as an inhibitor for DNA and RNA. I'm going to discuss DNA first and the pathway. So when it enters a cell, it can be converted to metabolites, 3 different metabolites. Fluorodeoxyuridine monophosphate, or FDUMP, fluorodeoxyuridine triphosphate, or FDUTP, or fluorouridine triphosphate, or FUTP. 5-FU, as it's called once it enters the cell, is converted into FDUMP and forms a complex with the thymidylate synthase. It inhibits the deoxythymidine monophosphate, or DTMP, production. And so because it inhibits the production and creates a low amount, DTMP is needed for DNA replication. So when there's low amounts, that cannot occur, and so that leads to cell death. So another pathway is the DPD is used to convert 5-FU to dihydrofluorouracil, and it's a rate-limiting step for the 5-FU catabolism for normal and cancerous cells. TS is catalyzes the methylation of deoxyuridine monophosphate, DUMP, to DTMP. And the incorporation of FDUTP into DNA inhibits TS by FDUMP, and it results into DNA damage. So, acting as a RNA inhibitor, it converts the conversion of F5U to 5-FUTP occurs. It incorporates itself into the RNA synthesis in the place of the uridine triphosphate, and it incorporates itself into RNA at a higher rate than DNA. Our RNA maturation is a more important target for F5U. So to further discuss the pathways of F5U. So once it enters the cell, it's converted into FUMP. It phosphorylates into FUDP, and it either is converted into FUTP, which then is when there's RNA damage, or FDUDP, which then leads to DNA damage within the nucleus once it's converted into FDUTP. So another pathway is the conversion of FDUMP, which occurs inside the nucleus. Thymidine synthase works by catalyzing the methylation of DUMP into DTMP. So the binding of FDUMP blocks DUMP and inhibits DTMP synthesis. And so that creates low levels of DTTP, which then lead to DNA damage and inhibits DNA synthesis from happening. So then here are my references.